Hi everyone, today let us see about bioprocess development, which is an interdisciplinary challenge. So, what is first bioprocess or bioprocessing? This is an essential part of many food, chemical, and pharmaceutical industries. Okay, these bioprocess operations make use of microbial, animal, and plant cells and components of cells such as enzymes to manufacture new products and destroy the harmful waste. These microorganisms, that is the what are the microorganisms you are using in the lab, it transforms biological materials for protection of fermented foods has origin in antiquity. Okay, since then bioprocess have been developed for an enormous range of commercial product. That is, many commercial products can be produced with the help of bioprocess. It may be an antibiotic or alcohol or therapeutic proteins, vaccines. These antibiotics, therapeutic proteins, and vaccines are expensive one. And so, from a low cost material like alcohol and organic solvents to high cost materials like antibiotics, therapeutic proteins, and vaccines can be produced with the help of this bioprocess for production, that is with the help of fermenter we are going to produce all these solvents or chemicals. So this process is called as bioprocess. The principles being used in this bioprocess development is called as bioprocess principles. Okay, and industrially useful enzymes and living cells such as bakers, brewers, yeast are also some of the commercial products used in bioprocessing. So we can see some of the commercial products which are being produced by this bioprocess development or bioprocess principles. Let us see first that. Uh, these are some of the commercial products. Here you can see fermented products like bulk organics. It may be ethanol or acetone or butanol. For ethanol production, you are using Saccharomyces cerevisiae, and acetone or butanol can be produced by Clostridium acetobotulicum. So the next column describes the approximate world market size. It may be 2 into 10 to the power 10 kilogram per year in the case of ethanol. In the case of butanol, you can see 2 into 10 to the power 6 kilogram per year. So this is one of the important product produced with the help of fermenter or bioprocess principles procedure. Then you can see biomass. It may be single cell protein or starter culture, yeast for food and agriculture. So that things come under biomass category. So the organism used can be lactic acid bacteria or baker's yeast. Single cell protein, that is become common nowadays. Uh, it may be Pseudomonas bacterotrophus or Candida utilis. Then organic acids you can see citric acid, gluconic acid, lactic acid, itaconic acid, citric acid from Aspergillus niger, gluconic acid from Aspergillus niger, lactic acid from Lactobacillus delbrueckii, itaconic acid from Aspergillus itaconius, and amino acids like L-glutamic acid from Cornybacterium glutamicum, L-lysine from Brevibacterium flavum, L-phenylalanine from Cornybacterium glutamicum, L-arginine from Brevibacterium flavum, and some other species like Cornybacterium also used to produce many amino acids. As you know, 20 amino acids are the main amino acids. Each amino acids can be produced by using some of the organisms. And microbial transformations, that is characteristics change. It may be steroids. Most commonly steroids are produced with the help of microbial transformation process, whereas you can use this rhizophus aureus d-sorbitol to L-sorbus. Here you can use acetobacter suboxidants, and a market value you can see 4 into 10 to the power 7 kilogram per year. And antibiotics, this is a very important market. Antibiotic, this is very important market. The antibiotics like penicillin from Penicillium chrysogenum, cephalosporin from Cephalosporium acrimonium, tetracyclines, macrolide antibiotics, polypeptide antibiotics, aminoglycoside antibiotics, aromatic antibiotics. The organisms used are Streptomyces aerifaciens, Streptomyces erytheus, Bacillus brevis, Streptomyces gracious, and Penicillium griseofulvum. I can see the market value also the next column. Some of the polysaccharide, extracellular polysaccharides like xanthan gum by Xanthomonas campestris, dextran from Lyconisma nestorites. Uh, so these are also some of the very important products produced with the help of fermentation technology. And enzymes like proteases, alpha amylase, glucoamylase, glucose isomerase, pectinase, rennin, all these are some of the enzymes produced. Vitamins like B12, riboflavin, ergot alkaloids, pigments like chiconin, beta carotene, vaccines, diphtheria for diphtheria disease, tetanus, pertussis, poliomyelitis, rubella, hepatitis. So these vaccines also can be produced with the help of fermentation technology. And therapeutic proteins like insulin, growth hormone, erythropoietin, factor 8C, then tissue plasminogen activator, interferon alpha 2, monoclonal antibodies from hybridoma cells, insecticides like bacterial spores, that is Bacillus thuringiensis, fungal spores, Histola thomposoni. So these are also can be developed with the help of fermentation technology. Here you can see the steps in development of a complete bioprocess for commercial manufacture of a new recombinant DNA derived product. Usually in a lab, you are using a microorganism which is being a recombinant one. The recombinant step you can see in the first one, you are using some biochemical or animal tissue, part of animal chromosome you have to take. You can cut and replace the chromosome with a microorganism. The fifth number you can see microorganism that is here. Microorganisms such as E. coli. From that you have to collect the plasmid, and from the plasmid with the help of restrictions enzymes you can cut certain part, and you can have the cut plasmid. In the cut plasmid you have to insert the part of a part or gene of interest, which gene you have to get the protein, or which gene it has to be expressed. That one can be restricted, and it can be ligated with the cut plasmid from a microorganism such as E. coli with the help of a ligase enzyme. So you are getting a recombinant plasmid here. This recombinant plasmid can be inserted in one microorganism for multiplication. So you are getting plasmid multiplication and gene expression here. So it divides, cell divides, divides and divides. You are getting mass number of cells, and you can go for small scale culture. After culturing the microorganism in small scale setup, you can go for the large scale setup. That is, you can scale up the culture setup. So you are going to benchtop bioreactor. Again, it can be pilot scale bioreactor. You can transfer it. After that, you can go for industrial scale production. That is, before industrial scale operation, everything has to be done in a pilot scale bioreactor. So in lab, after multiplication step, you are going for small scale culture. Then a benchtop is done. Then again a scale up is done. That is pilot scale bioreactor, which is more preferred in industries before going for industrial scale operation. Then product is being recovered, and then you can go for packaging and marketing. And also an important step is here, the waste products or effluents produced here has to be removed without creating any problems to environment. So these are all the steps usually involved in the fermentation industry to produce a product. Hope you all understand the video. Kindly like, share, comment and subscribe the video. Thank you.